Martinez. I'm part of the Department of Biological Sciences at Florida State University, and I conduct research investigating how cells and cell tissues interact with surfaces called polyelectrolyte multilayers. Those are thin film surfaces that have unique properties to them, in which we're trying to analyze, depending on what chemistry and properties we put into the films, if they promote cellular adhesion, motilities, or prevent it. The reason for that is you can use these thin films for biomedical applications in the future to promote better wound healing, assimilations with, implant sites, and maybe antibacterial surfaces. In particular, how I took the image for our STEM research event, where we're showcasing images of students that they took during research to kind of bring the attention of the public to STEM and learn more about it, is using this setup, which is an epifluorescent microscope. So the specific type of microscope that I'm using is called a Nikon Eclipse TI. So what's unique about that is that it allows me to see my cells both in regular light conditions and in a different condition called epifluorescence or fluorescent imaging. So what's unique about that is that I can actually target and look at specific proteins of the cells using fluorescent probes, or basically probes that when you hit them with a certain wavelength will emit a specific color. And that lets me look inside the cell and look at details for that. So I'm going to actually show you how that actually works. And so what we do is, usually when you're doing microscopy, the way I did it for the images for the Art and STEM project is you create a permanent mount. So basically you have your cover slip, your microscope slide, your cells are in between the cover slip and the microscope slide, and we're actually going to visualize that. Okay, so what I'm showing you today are just single cells that have been stained or probed to look at the nucleus. Actin, which is an important protein in the cell that helps keep the structure of it. So the color of the nuclei will appear as blue to me, and the color of the actin proteins, the cytoskeletal structural protein, will appear in green. So when we put the sample down, basically your specimen is facing towards the objectives of the microscope, the lenses that are helping us magnify the cells. So we put it down, everything's hooked up, and we use the Nikon software to actually help us capture the image. So we have a camera here that's looking at what I'm seeing with my eyepieces on the microscope, and you actually get to see your cells. So on here on my screen, you can see all the little individual fibroblast cells. And for the image that I took, it was actually of an epithelial cell sheet, but the process is the same of how you would capture the image. And I also investigate how individual cells like these fibroblasts and tissues migrate on our surfaces. So here they are, and then I want to look at how the nucleus, which I probed in blue, so I'm changing just the shutter systems here. And you can actually see the nucleus on here of all those little cells that we were looking at when they come out in blue. And then we can also look at actin, the structural proteins that keeps the shape of the cells together, and they appear in green. And then how I do it is you actually have to take two pictures. You have to take the blue picture first, the green picture, and then with the software you can put them all together.